Hello and welcome to this review lecture on lecture 17, 18 and 19. In this lecture we had done basic constitutional details about the deoxyribonucleic acids DNAs, and then we studied about different aspects of how to amplify the DNA in vitro, what are the proofreading mechanisms which are there for the enzyme for extending the DNA. As you know, PCR is a process where there are three different steps of different carried out at different temperatures. 95 degrees Celsius corresponding to denaturation of the DNA, 54 degrees corresponding to the annealing or the primer binding step of the DNA, and finally 72 degrees step which corresponds to the extension of the DNA molecule. And in respect of that, we also studied how to take a real time snapshot of the reaction through some fluorescence based or optical based method, where there would be a signal transduction mechanism. As the molecule grows, number of copies grow within the solution, there is a growth in the fluorescent signal, and that gives you an illustration of how the PCR process can be carried out in various stages along its amplification cycle. We know that there is a stage which corresponds to the first 5 or 6 cycles, where there would be a linear growth in the number of copies because of several diffusion limitations within the solution. There is again another stage where there would be an exponential growth, and then finally a plateauing stage which would indicate that the reactants, the primers, the enzyme, the DNTPs and the other constituents which are there apart from the template in the PCR reaction, they fall down, the concentrations of these fall down, because of which there is plateauing. So that's indicative of a real snapshot of the PCR process, and this can be very well gauged by looking at the fluorescence response. There are several assays which we discussed. One is the CyberGreen based detection, where there is a molecule CyberGreen which can intercalate with the double stranded DNA and result in a higher quantum yield. There is also another assay where we use a TACMAN probe of 2 different dye pairs and carry out a mechanism called FRET, fluorescence resonance energy transfer, where the 2 probes are bound to a primer before the primer binds to the template DNA, and the moment there is a binding of the primer to the template DNA and an enzymative activity, one of the probes or one of the molecular probe or a molecular dye, it comes out of the primer, and the 2 probes separate through a distance more than Forster's distance, and there is no resonance between the 2 molecules, and therefore their spectrofluorometric response changes, which gives you with certainty information about the right binding event or the right template DNA being amplified. We also further studied about a molecular beacon technology, where there would be a hairpin loop DNA acting as a primer, and it would be immobilized with a reporter and a quencher dye which would separate, thus leading to enhancement of the fluorescence as the primer binding event would happen. So these are some of the real time mechanisms we studied for the PCR process. We tried to take the PCR reaction to a microchip scale and used some basic thermal capacitance, thermal resistance based modeling, where we would take a mass of the PCR fluid and we would study what is the heat capacity and heat capacitance or thermal capacitance, and we would also study what is the thermal resistance. So this would be on a equivalent model, just like an electrical circuit, where the cause really is the voltage and the effect is a flow of charge. In a similar manner, in a thermal circuit, the cause is the temperature difference and the effect is the heat flow. So we considered that equivalence and tried to derive the thermal resistance and thermal capacitance, and finally put them, embedded them together into an RC circuit and tried to find out the response of the RC circuit, just in a way an RC circuit behaves, that the capacitor charges to its full value in about close to 3 times the time constant, a time equivalent equal to 3 times the time constant, which is also the product of the resistance capacitance product. So in the same manner, we have the thermal resistance, thermal capacitance product, and we multiply that with 3 to find out what is the ramp up time for the temperature to go to a certain preset value. In case of a PCR, as you know, these values are 95, 54 and 72 degrees Celsius. So, therefore, whenever there is a small confined volume which you are heating up and there is a small thermal mass which you are heating up, the way that it would go is that in one cycle of the PCR there would be at least 3 such ramp ups and ramp downs. Ramp down, the first ramp down would take place from 95 to 54 degrees, the second ramp up from 54 to 72 degrees and the third again from 72 to 95 degrees. So, in one particular cycle of the PCR we would have 9 times RC as the total ramping temperature of that small thermal volume, and this is purely dependent on the thickness. So, as we noted down that if you can take the thickness of the volume by making a thin film rather than a volume. Okay. Over and spread it over a huge surface area, obviously the amount of time needed to ramp up would be very small. It is proportional in general to the square of the depth of the film or the thickness of the film. So, therefore, film PCRs, which would be easily carried out in a microfabricated scale or microchip scale, would be much better in terms of its thermal its thermal ramp up, ramp down nature. So, it would have extreme rapidity because of very less such times. So, we also looked at various mechanisms to transport the DNA being negatively charged. Electrophoresis is one of the driving mechanisms for floating the DNA through gelatinous medium. There, as you may recall, we talked about a model where we would have the DNA charge being shielded by the equivalent positive charge of the medium, and this shielded charge would again transport across the gel, and then there would be a spherical capacitance module the electrophoretic mobility, and that mobility would again be a function of the field, and therefore, the velocity of flow of the DNA species would be proportional to the mobility, the electrophoretic mobility, which is very well dependent on the dielectric constant of the medium, and also the total amount of electric field which is present. So, we modeled electrophoresis, and then we tried to look at some of the different aspects of genomic detection, where we would simply put a PCR reaction at a chip scale and then pack gel samples within small capillaries and be able to translate the DNA fragments of different lengths as amplified by the PCR reaction through these capillaries. So, it becomes essentially a sieving problem, and we could be able to detect the various length strands at a very high throughput by making all sort of radial disc like arrangements, and there was some review from the literature which was carried out throughout these 3 lectures for indicating that. So, that in a way brings us to the end of the review of lectures 17 to 19. Thank you.